Bacteriophages, also called as the phages. These are the viruses which have the ability to infect the bacteria. As we know that there are different types of phages that are able to cause infections not only in humans but also have the ability to cause the diseases in the animals as well as the plants. So apart from this animals and plants, there are viruses which have the ability to cause infection in the bacteria, and these viruses which are exclusively the agents of causation of disease in the bacteria are called as the bacteriophages. So it was first described by the Toart when he has seen the degenerative colonies of the staphylococci, and when he used the culture filtrate, he was able to transmit the infection to the respective or the other staphylococcal colonies. It was carried out in 1915, and in 1917 similar type of experiment was carried out by D. Herrell, where he used the filtrate from the feces of a dysentery patient, and he used this filtrate to and he added the filtrate to the test tube containing the dysentery bacilli, and he could see the lysis of the bacilli, and he told that the lysis is because of the lytic agent which are the viruses, and hence he give them or coined the term as bacteriophages. And these are the, as we know that these are the organisms or the viruses which have the ability to cause the lysis of the bacteria. These phages are usually in close association with the different types of bacteria, and hence can be easily isolated from the feces or the natural severages, etc. Initially it was thought that because these viruses have the ability to kill the bacteria, they can be used for the treatment of different bacterial diseases. Still the work is going on in that particular area, but these viruses has given a lot of scope to this virology for studying and making the bacteriophages typing or the typing of the bacteria for the epidemic purposes to identify the bacteria that is usually involved in the causation of the disease in that particular areas. Now, these organisms can be easily grown on the bacterial culture, and hence it helps us to know the viral host interaction, not only at the molecular level but at the submolecular level too. These viruses have the ability to transfer the genetic information from one cell to the other cell by a process which is called as a transduction, which is a genetic recombination. As we know, recombination is of three types, that is conjugation, transformation and transduction. So the genetic information transferred or carried by the phages and passed on to the other cell is called as a transduction. The recombination mediated by the phages is called as a transduction. And these phages have the ability to get or release their viral nucleic acid into the host cell, and the nucleic acid has the ability to get integrated into the host cell chromosome, so making a phage conversion. So here the phage nucleic acid will carry certain types of genes which confer certain property to the host cell. So this is called as a phage conversion. The best example of this is the diphtheria bacilli. Diphtheria bacilli is itself not toxigenic in nature, but when it is lysogenized with the help of bacteriophages or beta bacteriophage, the beta bacteriophage will carry the toxin gene. Now when it is lysogenized, the diphtheria bacilli have the ability to produce the toxin. So that is called as a phage conversion. The integration of the viral nucleic acid into the host chromosome makes the host phage conversion and gives certain important property to the host cell. And next important property of this or the function of the bacteriophage is that they can be used as a cloning vectors or a cloning vehicles in recombinant DNA technology or genetic manipulations. And because these organisms are able to kill the bacteria, they can be thought of control to the bacterial population in the natural environment. And because of the host specificity with the bacteria, that is any viruses cannot affect any type of bacteria. There is a host specificity. There are certain viruses which can infect only certain type of bacteria. Now based upon this logic, we can make use of this phages to identify the bacteria and type the bacteria, which is called as a phage typing, helps to identify the bacteria which is causing a particular disease. Coming to the morphology of this bacteriophages, it was first discovered in E. coli, and these are called as a T event phages, which has been studied in detail, which gives us all report about how the bacteriophages usually looks alike. And these bacteriophages usually have a complex morphology and a tadpole shaped structure. They have a hexagonal head into which is a packed nucleic acid which is in the form of a DNA, and they have a cylindrical tail through which the nucleic acid usually enters into the host cell. The head is filled with the double-stranded DNA molecule, and the tail is a cylindrical hollow tube consisting of a contractile sheath, has the ability to contract and expand when it comes in contact with the host cell. The tail usually ends with a base plate ending up with the tail fibers. The tail fibers are very important to identify receptors on the bacterial surface. So it has a complex structure with a phage head and a tail ending up with a base plate and the prongs or the tail fibers. So certain bacteriophages can have fibers or the prongs. And apart from this complex structure, there are certain bacteriophages that also exhibit spherical or filamentous structures consisting of single-stranded RNA or the DNA. So it is either the DNA or RNA that can be present as a genetic material, but not both the things. So when the DNA and RNA is present, DNA only acts as a genetic material. So there are certain viruses that carry RNA as a genetic material too. Coming to the life cycle of this organism, it is an interesting part of the bacteriophage. The bacteriophage exhibits two types of life cycle. One is called as a lytic cycle, where there is an intracellular replication of the progeny virus, which are usually produced inside the host cell, and they usually kill the host cell and comes out of the host cell. This procedure is called as a lytic cycle, and the other alternative form of replicative cycle is called as a lysogenic cycle, where the DNA of the viral nucleic acid usually gets integrated into the host chromosome and lives in harmony with the host chromosome. It synchronously replicates as and when the host chromosome replicates without causing any harm to the host cell. So this type of life cycle is called as a lysogenic life cycle. Coming to the life cycle in detail, we'll deal with the lytic cycle. There are different steps that are involved in the lytic cycle. First is the adsorption, next is the penetration and synthesis of the viral compounds. Next is the assembly of the all the compounds, that is all the particles of the phages, that is the head, tail, nucleic acid, etc. are synthesized differently but not together, and then they are usually packed to form assembly, and then the whole virion usually matures. The infecting unit of a virus is called as a virion. So it is only after the maturation, after assembly, maturation takes place, and it usually forms a virion which has the ability to now infect the other bacteria. Now once the maturation takes place, all the progeny viruses have been synthesized, then there is release of the progeny, which is by the lysis of the host cell. So this all are the steps that are included under the lytic cycle. So we shall discuss this in detail. Let us study the slide first, the picture that helps us to explain how the lytic cycle usually takes place. So you can see the bacteriophage attaching to the host cell with the help of tail fibers, and then with the help of the contractile sheath, it inserts it or releases its nucleic acid, that is the DNA, into the host cell. Now once the DNA enters into the host cell, it completely takes over the host cell and synthesizes its progeny particles separately. That is, the head is synthesized separately, tail is synthesized separately. We have DNA being synthesized and other protein components of the viruses being synthesized separately. Once all the components are synthesized, they usually assemble together. First, the head, into the head is the insertion of the DNA, after which the tail assembly takes place, and the tail fibers, base place, and the tail fibers takes place. So once a complete virion particle is formed, all the daughter particles are formed, they are released by lysis of the host cell. So this slide very well explains how the lytic cycle usually takes place in the bacteriophages and into the bacterial cell. Let us study this in detail, each step in detail. Coming to the adsorption, adsorption is a rapid collision between the phages and the bacteria. So when there is a collision between the phages and the bacteria, the phage particle usually serves the surface of the bacteria with the help of the tail fiber, and then attaches to the host cell receptor and carries on the further processes. So it is a tail that helps in identifying the chemical receptors that are present on the host cell. And there is a specificity between the attachment of the tail and the host cell. As already discussed, any bacteria, any cannot be infected by any virus. There are certain viruses that are specific towards the bacteria because they have got this chemical receptor that are expressed on the surface that are recognized by the tail receptors. So when there is an interaction between the tail receptor and the bacterial surface receptor, then there is an adsorption, and under optimum condition the adsorption is usually rapid in nature, taking about minutes. And apart from this, the cofactors are also required to carry on the adsorption process. The cofactors are usually in the form of cations, and the bacterial cell wall expresses chemical receptors that helps in identifying the viral surface receptors, and attachment takes place between these two receptors. Now in case of certain bacteria who do not have a cell wall, which are called as a protoplast, or the bacteria which have lost the cell wall, in that case the receptors are lost, and hence the protoplast cannot be infected with the bacteriophages, and hence they are immune towards the bacteriophages. But this property can be regenerated when the cell wall is added to the protoplast, or the host specificity can be carried out by to the host cell, that is a bacterial cell can be infected even though there is no specificity. So this procedure is called as a transfection, where a naked phage DNA can be allowed to enter into the bacterial host cell by using different types of chemicals. So this is called as a transinfection phenomena. Now coming to the penetration, once adsorption takes place, that is a random collision between the host cell and the viruses with the help of receptor, there is an attachment. Now next is the penetration of the nucleic acid into the host cell. So the nucleic acid is the one which is infectious in nature, and it is not the head or the tail. So once the nucleic acid enters into the host cell, it becomes pathogenic and causes the lysis of the host cell. So the penetration of the nucleic acid into the host cell is similar to that of the injection procedure. The base plate and the tail fibers holds the bacterial surface tightly, and the contractile sheath usually enters into the host cell with the help of phase tail lysozyme that causes the pores on the surface of the bacteria. Once the tail sheath is inside, it releases the nucleic acid. So in order to carry out this particular function, it requires a small amount of adenosine triphosphate, that is ATP, which is derived from the tail fiber. So once the contractile sheath enters into the host cell, there is a release of the phage nucleic acid, and the phage DNA enters into the host cell. The remaining part that is left outside, that is the head and the tail, which does not have a DNA, is now called as a ghost. Sometimes it so can happen that the bacterial cell can be infected with the different types of phages, that is different phages, but these phages do not carry out the replicative cycle, but instead they can kill the bacteria by releasing the cell contents. So this phenomena is called as lysis from without. So there is no replication taking place in the bacteria, just the leakage of the content. They infect the bacteria, cause the leakage of the content and death of the bacteria. So that phenomena is called as a lysis from without. Now the third important step is synthesis of the phage component. So here first we have adsorption, where the virus has adsorbed onto the host cell by recognizing specific receptors. Then there is introduction of the nucleic acid from the contractile sheath, and then the assembly, the synthesis of the phage components. So for the synthesis of the phage components, as already described, each component is synthesized separately. So in order to synthesize that, we need different types of protein. So first proteins that are synthesized when the viral DNA enters into the host cell is the early protein. Early proteins are the one that helps in synthesizing the complex structures, whereas the late proteins are the one that synthesizes the protein subunits such as the head, tail fibers, etc. Now once the DNA enters, it has to take over the host cell and use RNA polymerase to synthesize the viral particles. So once the viral DNA enters, it seizes all the bacterial proteins, all the bacterial metabolic activities, and take over the metabolic activities and synthesize its components rather than the bacterial components. So now everything is under the control of the viral nucleic acid. So once the early proteins and late proteins are synthesized, phage DNA, phage head protein, tail proteins, etc. are synthesized. Now once all these are synthesized separately, it is the head into which the DNA is first packed till the head completely fills with the help of the DNA. Next, the assembly takes place is with the help of the tail structures. That is, the tail contractile sheath or the hollow fiber attaches to the head, then the base plate and the tail fiber. So this is how the assembly completely takes place. Now once the mature particle is formed, it is called, the phenomena is called as a maturation, and this particle is now called as a virion because it has ability to infect the other cells. So similarly, there are thousands of virion particles that are formed inside the bacterial cell. Next is the release. Once all the daughter progenies are formed inside the bacterial cell, now next is to lyse the bacterial cell and get released to infect the other cell. So during the process of the synthesis of these virion particles, the cell wall of the bacteria gets weakened, and it completely changes its structure from the ellipse stage to the circular stage, and it becomes weakened due to the phage enzymes that are usually produced during the replication process. Once the virion particles are formed, the phage enzyme usually ruptures the host cell and causes the release of the daughter phages. So this step is called as the release. Now once the virion are out, they are ready to infect the other bacteriophages or the bacteria. Now there are certain terms that have to be described in lytic cycle. One is called as the eclipse phase. The phase between the entry of the viral DNA and the appearance of the first progeny virus is called as the eclipse phase. Next is the latent phase. The phase between the entry of the viral DNA and the release of the first progeny virus is called as the latent period. Now rise period is as soon as there is a latent period when the daughter cells are usually released. Initially there is an increase in the release of more amount of the daughter DNA molecule or daughter progeny molecule. So this period where you can see a heavy release of the daughter progeny phages is called as a rise period. Now the burst size represents the average number of daughter progeny viruses that are produced per bacterial cell is called as a burst size. This can be very well explained with the help of taking one bacteria, infecting it with a single phages, and seeing the number of or estimating the number of progeny phages that are released and plotting against a graph. So this graph is called as a one-step growth curve that helps us to know the burst size, that is how many phages can infect the bacteria and how many progeny viruses are usually produced from this bacterial DNA. Now next important life cycle is a lysogenic cycle, lytic cycle, where there is a lysis of the bacterial cell or killing of the bacterial cell usually takes place. Whereas in the lysogenic cycle, the viral DNA which one enters into the host cell, the DNA gets integrated into the host cell without causing the lysis of the host cell. So these phages are called as the temperate phages, and there is a symbiotic relationship between the viral nucleic acid and the host nucleic acid, and it works in synchrony with the host cell and replicates as and when the host cell replicates. So the viral DNA which gets integrated into the host DNA is called as a prophage, and the cycle is called as a lysogeny, and the bacterium that harbors this prophage is called as a lysogenic bacterium. Now this as already discussed, the viral DNA sometimes can carry important genes that confers important property to the host cell. So this particular phenomena is called as a lysogenic conversion or the phage conversion. So described earlier with the help of diphtheria toxin. Now sometimes it so happens that the lysogenized DNA can get excised from the host DNA during unfavorable conditions. So this step is called as the excision. Once excised from the host DNA, it now has the ability to carry out the lytic cycle and cause the lysis of the host cell and get released from the host cell. So this usually occurs when there is a spontaneous induction of the phage either due to physical agents or the chemical agents. The physical agents such as UV. So when the organism is a threat under UV conditions, the prophage is the first that usually comes out, causes the lysis and gets released and infects the other bacteria. Similarly, we have chemical agents such as nitrogen mustard, hydrogen peroxide, etc. These are the chemical agents that can induce the lytic cycle from the lysogenic cycle. Now, when a bacteria is infected with a particular temperate phase, that is a lysogenized with a temperate phase, it cannot be infected by the other bacteria, other phases. So, it is called as super infection immunity. So, once lysogenized, it is immune to different types of phases. It does not get infection from the other phases or cannot be infected with the other phases. So, that term is called as super infection immunity. Now, once the organism or the phases get integrated into the host cell, they will have the ability to transfer the genes from the host chromosome to the other cell. So, this phenomena is called as a transduction. So, transduction is of two types. One is called as a generalized transduction, the other one is called as a restricted or specialized transduction. So, in the restricted transduction, the name itself indicates that it is restricted, that it carries only particular genes from the host chromosome and transferred to the other host. So, this usually takes place when the lysogenic cycle is usually carried out. So, when the DNA gets integrated in the host cell, during the excision time, it so happens that it leaves the phage DNA leaves some of its genes and carries some genes from the host chromosome. Take for example, in the E. coli K12 strains, when the host cell is lysogenized with the temperate phase, the temperate phage or the nucleic acid get integrated only at a space between gal and biogenes. So during the excision, it so happens that the viral DNA leaves its nucleus and nucleic acid and takes part of gal plus genes, and now when it infects the other bacteria, it will have the ability to transfer this gal plus gene. So that particular phenomena is called as restricted or specialized transduction, usually carried out by only the temperate phase. Whereas the generalized transduction is seen during the lytic cycle, because as soon as the viral DNA enters, it causes the lysis of the host DNA. So during maturation, it so happens that some of the host DNA can get accommodated in the head and can transfer the information to the other host cell. So because there is no specific gene that get integrated into the head, it is called as a generalized transduction. And this phenomena is called as a genetic exchange that helps in the production of the recombinants. And a similar type of resistance can be seen in certain staphylococcal colonies, where the plasmid mediated drug resistance can be very well explained. The plasmid carrying the resistant gene can be transferred from the host cell to the other cell, and making the other cell also resistant towards the different types of antibiotics. When single bacterial cells get infected with different types of phages, they have the ability to multiply, and during the multiplication, it so happens that recombinants can be formed, and some of the daughters which are recombinant in nature can be released, giving rise to a new type of recombinant strains. Now, how do you estimate, how do you identify the bacteriophages on a bacteria? So, the bacteria are usually killed by a process called as lysis, when the virus usually attacks a bacteria. So, this can be very well seen on a media, where the bacteriophage which insects the colonies of the bacteria causes a lysis, resulting in the clearing zone, which are called as a plaques. So, the number of plaques represents the number of the bacteriophages that are infected and cause the lysis. So, clearing of the zone is seen only when there is a lysis of the colonies, and the size, shape and the structure of the plaque can determine the type of the bacteriophage that has usually infected the bacteria. And this can be used to titrate number of the viable phages, and because it is analogous to the bacterial colonies, this can be used in the purification of the phages too. Next is the bacteriophage typing, very important in identifying the type of bacteria. Now, as we know, there is a host specificity between the viruses and the bacteria, and only certain type of viruses can infect certain type of bacteria. This helps in identifying the bacteria. So, this is called as a phage typing. So, this is because of the phage bacterium interaction and host specificity. There are certain phages that have broad specificity, broad host specificity, whereas certain have narrow host specificity. The bacteria, the phages with the broad host specificity are the one that can infect most type of bacterial colonies or the most type of bacterial species, whereas the narrow host specificity bacteriophages can infect only particular group of bacteria. So these are mostly used in bacteriophage typing, identifying the bacterial agent involved in the disease causation. Now sometimes this bacteria can be made to get adapted to the phages by successively cultivating the viruses in the bacteria, which are not specific initially. Later on they become specific, and this particular phenomena is called as the adaptation of the host cell. That is, the host get adapted when the phages are serially cultured or serially infected with the same type of phages to the bacteria. Now there are certain phages which are genus specific that can cause the lysis, that can infect a large group of genus, for example Salmonella, and species specific that can infect only the specific species of a bacteria such as Bacillus anthracis. And there are certain phages which are called as biotype or subspecies phages, which can infect only particular subspecies in a organism, that is a genus. Take for example Vibrio cholera. They have two types of organism, classical and Eltor. So there are subspecies bacteriophages that have the ability to infect only the classical V. cholera. And this phase typing is mostly used for the intra-species typing. For example, to type Staphylococci and the Salmonella typhi. So intra-species classification can be done with the help of bacteriophage typing. And there are adapted phages that have the ability to infect only the fresh isolates of the VI antigen, and hence in typing the typhoid bacilli. And coming to the staphylococcal phage typing, it is mostly used to identify or classify, give nomenclature to the type or the number of the phages that have the ability to infect the staphylococci species. So this can be carried out in an experiment where a strain of staphylococci is infected with a number of phages, and the phage type can be can be given the name depending upon the number of phages that has infected the staphylococci. Now, as we know that the lytic property is based upon the dose of infection. So before making use of this phage as for the bacteriophage typing, you have to standardize it by titration. So titration is usually done by serial dilution of the phage, which are then added to the colonies or made to lyse the colonies or lawn of cultures, after which the lawn of culture are usually incubated. The highest dilution of the phage that have ability to produce a confluent lysis is taken as a routine test dose or RTT. Next, similar structures that are related to the bacteriophages are the bacteriocins. Though they are not bacteriophages, but these show chemical properties or similarity with the bacteriophages. So they are called as bacteriocins. It was first isolated by Gracia from E. coli organism. So colicins are nothing but the antibiotic substances or certain toxic substances produced by one group of organism that has inhibitory effect on the closely related group of organism. That is, E. coli organism produces colicin that have the ability to kill the closely related group of E. coli organism, and this is the one that helps the E. coli organism to survive in an environment where there are number of different organisms. So it was first isolated by Gracia, and the term was given as colicin. Similar type of substances were seen to be produced by different types of bacteria, and hence a common name is given to them, which is called as a bacteriocin. And specific names are given depending upon the type of colicin produced by a type of bacteria. Take for example pyocin produced by Pseudomonas pyocyaneae, and megacin produced by Bacillus megaterium, and these are produced mostly by the organism, and the chemical property of this is they are proteinaceous in nature, and sometimes can be lipopolysaccharide, which is a component of the gram-negative cell wall. And this coalescence usually resembles the phages in their activity. That is how they usually bring about the lysis of the host cell. The similar type of activity can be seen in the bacteriocins. And Piocene have been studied, that is from the Pseudomonas piocinae. The Piocene have been studied in detail, and they have seen that the Piocene resemble the tail structure of the bacteriophage, and hence they thought that it is a defective phage genome that was able to produce only a part of the bacteriophage, and it is a core factor that is responsible for producing the coalescence, and it is an episome that can be transmitted with the help of conjugation or transduction. Physical and chemical agents helps to enhance the activity of the bacteriocins. Now, the organism which produces this bacteriocin is immune to its own coalescence, but have the ability to infect and cause the lysis of the other organism. That is, it itself is immune to it. It is not killed by its own bacteriocin, but instead can kill the other organism. And it is highly specific in nature. The specificity can be very well understood when we type the bacteriocins. That is, bacteriocins cannot infect or cause the lysis of any bacteria. They are highly specific and they target only specific group of organism. Take for example Shigella soni, Proteus species, and Pseudomonas aeruginosa. So they are the sensitive cell towards the bacteriocin. And bacteriocin typing is similar to that of the phage typing, but in phage typing we will see the sensitivity of the strain of organism which is sensitive towards a particular phage. But here in the case of bacteriocin typing, we will see the type of bacteriocin produced by the test strain, which is usually inhibitory to the organism, that is the indicator organism. So this is usually done with the help of plate diffusion. In the plate diffusion technique, we make a broad streak of the test organism that is able to produce the bacteriocin. After the growth of the organism, it is completely scraped off, leaving behind certain colonies. Now these colonies are killed by the chloroform. Now the test strain is the one that has produced the bacteriocin. In order to identify the type of bacteriocin that has produced, in order to identify how it is able to cause a lysis of the indicator strain, the indicator strain is usually streaked at 90 degrees or the right angle to the or indicator strain and incubated. Now depending upon the type of infection that it shows on the organism, we can easily type the bacteriocin that is produced and the type of strain onto which is usually infective. So this is about the bacteriophages.